All right. Hello. I hope you're doing well, you're managing to stay healthy, and you're keeping on top of all the work your teachers have been assigning. So, the reason for this video is to help you get started with the next stage of our summative assessment. Now, I've already given you feedback on stages 2 and stages 3. So hopefully you saw that and you can use that feedback to make sure going forward you're going to do even better. Remember, everything that you submit on these stages, I'm using as evidence of what you know, what you understand, what you can do. So you want to do the best you possibly can on all these stages. That means writing thorough, concise, accurate responses, making sure that you've written responses to all the questions, that you're investing time into doing research and so on, right? All right, so stage 2, you took an individual kind of test to show if you understood the importance of proteins and specialized cells. Could you transcribe and translate a sequence? Could you mutate a sequence of DNA and so on? Stage 3, you did a little bit of research on DNA nanobots, codes, and the universal genetic code. Now, what you're going to do in stage 4 is you're going to look at the natural code and you're going to see how it responds to certain types of mutations. You're going to see how it responds to insertion mutations, substitution mutations, deletion mutations. And you're going to compare the effects that each of those mutations have on the sequence of amino acids. And you're going to do this with other group members. And I'll tell you your group members towards the end of this video, all right? But first, in Google Classroom, you should have a document that looks like this, right? And here, again, this is going to be your control. This is the amino acid sequence that you're going to work with and that you're going to mutate. Now, they're doing construction next door, so if there's a little bit of noise, I apologize. But here, okay, so you have this amino acid sequence, and remember, amino acid sequences start with methionine. You don't start transcribing until you see an AUG codon in the mRNA molecule, right? Okay, so we start with methionine, and then we go through a sequence of 1, 2, 3, 4, 5, 6, 7, 8 other amino acid sequences until we hit a stop codon. So what you're going to have to do in number 1 is you're going to have to work backwards. You're going to have to take this amino acid sequence and reverse translate it, right? You're going to try to figure out the mRNA codon sequence that was responsible for producing this amino acid sequence. Now, as you hopefully remember, you could have a few different codons, a few different mRNA codons that code for the exact same amino acid. That's okay, you just pick one that could have been responsible for coding for this amino acid sequence. So once you work backwards and have reverse translated and you have an mRNA sequence, then you're going to reverse transcribe that mRNA sequence into a DNA sequence. That DNA sequence then you're going to record in this particular table right here, right? That's where you have your original DNA strand. So you put your A, C, T, G sequence there. You're then going to mutate that DNA sequence following instructions that are right here. So for example, it says mutation 1. You're going to substitute every third base in the DNA sequence. So you're going to count 1, 2, 3 bases, and then you're going to mutate that particular base. So if that particular base is an adenine, if it's represented by the letter A, you're going to change that A to either a thymine T, a cytosine C, or a guanine G, right? And then you're going to count 3 more bases from that, and that third base, let's see, that third base is a C. Then you're going to change that cytosine to either a guanine, an adenine, or a thymine, and you keep doing that until the end of the DNA sequence. Once you have that mutation, you record that mutation where it says substitution mutation 1. You record that DNA sequence there. You're then going to transcribe that sequence into an mRNA transcript and then translate that sequence into a sequence of amino acids. Now remember, we do not start transcribing until we see a start codon, right? In DNA, if it's a sequence of TAC, if those are the bases, that's the start of the gene. In mRNA, if it's an AUG, we know that codes for the amino acid methionine, abbreviated as capital MET or MET. That's when we start translating, right? If we don't have a TAC in DNA, or if we don't have an AUG in our mRNA molecule, we don't translate. We can't because there's no start. So if that's the case, you have to look through the rest of the sequence until you see that sequence of letters, that sequence of bases before you start transcribing or translating. Okay? Now then, we go back to the original DNA strand, and then we do the second mutation, where it says substitute every fourth base. Again, we don't mutate the strand we already mutated. We mutate that original DNA sequence again. So we count 1, 2, 3, 4 bases. Maybe you highlight it, and you substitute that base for another base. Count another 4 bases, highlight it, substitute that base, and then you go through the same process. You transcribe and you translate that base, and you see, okay, what is the new amino acid sequence that is produced? Now, you see that little gray box where it says number of changes to amino acid sequence? That's where you compare the original amino acid sequence, that one that I have highlighted in red here, right? The methionine, alanine, proline, so on. You compare that to the sequence that you create after the substitution. You have to compare them one for one. It's not just, does the amino acid exist in that original sequence? It has to be in the exact same sequence. It has to be in the exact same order. So if you have a methionine for the very first amino acid, that's not a change, right? But let's say that for the substitution mutation 2, you translate your mRNA sequence and you get methionine and then a stop. If you get a methionine and then a stop, then there's only one similarity to the original sequence, right? The methionine that's at the very beginning. The stop is not what happens during the second amino acid. Instead, we have alanine in the original. So that means we would have 9 differences in our mutated sequence compared to our original sequence. And where we would have the number of changes to amino acid sequence for substitution mutation 2, we would record the number 9 there. So again, part of the goal is to look at how these different mutations affect the sequence of amino acids that eventually is translated to make a polypeptide sequence. You are going to compare all of your mutations here. You're going to get to the analysis questions, and again, working with your group members, you can respond to these questions together. You can compare, refine, but you want to do the best you can to show a thorough analysis that you understand what's going on for each of these four analysis questions in each of the 3 mutations. Now, again, you just follow for each of these mutations the directions that I have provided for you here for the deletion mutation, here for the insertion mutation. You're keeping on going back to this chart and you're recording. The original DNA sequence never changes, right? But the deletion, the transcribe, and the translate will change. And that's what you need to keep track of. All right. Now, again, you're going to be doing this in groups. And remember, again, I'm just going to highlight, I'm going to reiterate because this is important. You can't translate until you get a MET, right? You have to have an AUG. If you don't have an AUG, then you have to keep on looking at your sequence until there's an AUG before you start translating. Or again, if it's DNA, you're looking for that ACT. If you don't see an ACT, you don't start transcribing at all until you see that ACT. And once you see that, that's where you start. Now, you're working in groups, and I'm just going to use note cards to determine your groups. So I shuffled these, and here are your group members for stage 4 of this task, all right? So you are going to be working, with these few people. And let me just minimize this, and here we go. So first group that will be working together will be Emmanuel, Kieran, Sean, and Alex. All right, so you're responsible for working together. You should reach out to each other either on your WhatsApp chat, on Google Classroom, via email. Come up with a shared document and then work together on this, okay? Next group will be Jan, Michelle, Martin, and Aiden. All right, that's group 2. Group 3 will be Megan, Michael, Here, and Alexandra. And that means our final group consists of Mark, Eden, and Igor. Again, you're responsible for working together and getting stage 4 complete by the due date, and making sure one group member, one person from your group, uploads your completed document to the Google Classroom assignment so I can look at it and give you feedback by the due date. All right? I hope this makes sense and clarifies what you're responsible for. I'll try to be available via email or Google Classroom if you have any questions. And if we're out of school for much longer, hopefully we can video conference during maybe some set office hours on Zoom, which you will download in the very near future. All right? So stay healthy, and I hope to see you soon. Bye.